Hey, Nate,
Yes, Sam.
have you ever been to the top of Mount Nittany?
No.
Wait a minute. You've never been to the top of Mount Nittany?
No, I've never been to the top of Mount Nittany.
Now, you like walking around the woods, right?
Yes,
You know that it's like the thing to do when you live in this area, right?
I know. I'm sorry.
And more importantly, you know that we planned this as our cold opening, right?
Yes.
Just go along with me, okay?
Okay, gotcha.
Hey, Nate, have you ever been to the top of Mount Nittany?
Yes, I have actually.
Right. And when you're up there, did you ever hear something rustling around in the woods? Definitely sounds bigger than a squirrel.
It's probably a porcupine, but
Well, you're probably right, but I was wondering, could that be a Nittany lion?
wait. Were those a thing?
Did they even exist?
I don't know.
Hey,
I got a question about that. Welcome to the third episode of Hey, I Got a Question About That.
Right. It's a podcast and video series where we talk about all the really cool research that goes on here at the Eberly College of Science at Penn State.
Yeah, in this episode, we're talking with Shaun Mahony. He is an assistant professor of biochemistry and molecular biology here at Penn State. And he uses computational biology to study gene regulation.
Right. And we have another guest this time. Joining us via satellite all the way from Germany is George P.J. Perry. He's an associate professor of anthropology and biology at Penn State, and he studies anthropological genomics and ancient DNA.
So Shaun and PJ are actually working on a project to sequence the NITTANY lion genome.
Right. And the whole project is wrapped up in one of the courses Shaun teaches, so undergraduates are involved directly in the research.
Yeah, it's really neat to see how students have been involved in this project every step of the way.
Let's check it out. All right, so we're joined here in the studio by Shaun Mahony, who's an assistant professor of biochemistry and molecular biology. Thanks for joining us, Shaun. Can you tell us a little bit about your research?
Sure. So I'm a computational biologist. I study, I apply machine learning methods or develop machine learning methods for understanding how the genome works. So specifically, we work on understanding gene regulation. Every cell in your body has different sets of genes active. So we're trying to figure out what control switches regulate that process, like which switches turn on genes in different tissues in your body and different cell types in your body. We're particularly interested in understanding which switches you'd have to turn on or off to turn one cell type into another cell type. So if you're interested in making some particular type of neuron or pancreatic cell or something like that, how would you go about that from different cells, from skin cells or something like that?
Very cool. And we're also joined via satellite by George, or P.J. Perry. He is the Harry J. and Elissa M. Sichi Early Career Professor in Anthropology and an Associate Professor of Anthropology and Biology. And he's joining us from Germany. Thanks for joining us, P.J. Can you tell us a little bit about what you do?
Yeah, thanks so much for having me. I am an evolutionary genomicist, so I study genomes to help make inferences about the evolutionary history of a species or a population. And the species that I study are often humans, non-human primates, but also a lot of other species out there whose population history and evolutionary history has been affected by past human behavior. And we do this through a combination of methods, but it includes studying modern DNA, modern genomes, but also ancient DNA and ancient genomes. So the DNA from extinct species or DNA from bones and teeth of extinct populations of still living species, because that's a very powerful tool, and of course something that we're going to be talking about a little bit today.
Yeah, that is a perfect segue into what we're talking about, because Nate and I were having a conversation, and we were thinking about, here at Penn State, our mascot is the Nittany lion, and we kind of got into wondering, what is a Nittany lion? Is that an actual thing separate from other types of mountain lions? And I know you both are working on a project that is looking into that a bit. Can you tell us a little bit how you got into that?
Yeah, I think this is one for me to start, and then we can transition to Shaun in a minute. But because we have the ancient DNA lab at Penn State, and that's becoming an increasingly powerful tool for studying past population dynamics, we got interested in using the ancient DNA technology to do a study of Nittany lions, which, Nittany lions the Nittany lion is the mascot of Penn State, but there are not any lions anymore around the Penn State campus, but there used to be. And in fact, the Nittany lion name is named after the mountain lion that used to be in the region. But those mountain lions went extinct from this region in the late 1800s due to overhunting and habitat loss. But there are still some skins, preserved museum skins of the mountain lions that from this region that remain. And so we had the idea to use the Ancient DNA Lab to study the genomes of those individuals to help us understand what we lost with the regional extinction of the northeastern mountain lions, which we're calling Nittany lions.
And then, Shaun, you've incorporated this into a class that you're teaching, right?
PJ here got some funding, I guess, to sequence the Nittany lion. Crowdfunded that a couple of years ago. I'm sure you're going to tell them about that in a few minutes. But then he approached me about incorporating that data that he had generated into a class that I teach. So the class is Intro to Bioinformatics. It's BMB 482. This is a class that we've been running here at Penn State for the last 4 or 5 years. So it's a broad introduction to bioinformatics, first of all. So it's meant for upper level undergraduates. We get some graduate students taking it as well. We cover basic topics in bioinformatics. So bioinformatics, or computational biology, is the application of computational analysis methods to biological data broadly. But in our class, we focus a lot on DNA sequences and the genome. So we go through a lot of different aspects of how you compare DNA sequences against each other, how you build genomes out of a large collection of DNA sequences, so that's genome assembly, how you find out what parts of the genome mean different things. Are there genes, where are the genes on the genome, where are the control switches that I study on the genome? And then, of course, we look at broader questions of comparing genomes against each other. So that's where you get into evolutionary questions. So you can tell that there's a bunch of topics that we cover in this course that would be quite amenable for looking at a set of DNA that we've sequenced from something like the Nittany lion, and then asking some biological scientific questions about that sequence. How is it related to other cat species? How does it compare to other known sequences from other mountain lions? So this was a really nice opportunity for my class to get some practical experience over the last couple of years in applying bioinformatics methods to some data that PJ had generated.
I'm going to back up a little bit here. How do you handle contamination with these old skins, and what's some of the struggles you have working with ancient DNA?
Yeah, so ancient DNA is a major challenge because in our living bodies, our DNA is all strung together, very long strings of A's, T's, C's, and G's, which all together combine to give cells the information that they need to form our body parts and to do the processes, the biological processes that go on in our body. But after an organism dies, the long strands of DNA start to break apart. And over time, they break apart into very, very small pieces. And DNA from other sources, say bacteria, other people who have touched the specimen, and so on, start to also become mixed in with the sample. So we have a very specialized clean lab at Penn State where students who work in the lab, they put on clean suits. Everything is UV treated, and we're very, very careful about contamination. And we have a lot of methods to extract very small quantities of DNA. And then we use sequencing technology that can sequence small fragments of DNA specifically, because that's all that we have left for these specimens. And then third, we have specialized bioinformatics tools like what Shaun Shaun's class is covering, to be able to work with the massive amounts of data that we generate to be able to pull out the needle in a haystack that is ancient DNA that we're looking for from the NITTANY lion in this case.
We're talking about the NITTANY lion genome as ancient DNA, but we're talking decades old, right? Or maybe 100 years old.
150 years.
People have sequenced Neanderthal, and people have sequenced a woolly mammoth. How does working with this DNA that's new compared to that, compared to those other ancient genomes that have been done?
Yeah, I mean, this is a great question, and I'm calling this ancient DNA because we use our ancient DNA lab to study it. Other people would refer to these samples as historical DNA, and it doesn't really matter what you call it. What matters is that there's some shared properties in terms of what makes this challenging, but then of course also exciting, because here we have an opportunity to learn about the population history in biology of a population of an iconic animal that is now regionally extinct and happens to be the mascot of Penn State, and through which we can engage Penn State students in research and everything. So I think it's very worth the challenges, and it's very exciting, but it is very difficult. You mentioned that ancient DNA has been successfully extracted and studied from, for example, Neanderthals, which are a population somewhat distantly related to modern humans, but that also had some mixture with modern humans. And some of these samples, many of these samples are 40,000, 50,000 years old or older. Some of the Neanderthal DNA that's been sequenced is better preserved than some of the NITTANY lion samples that we're working with. And it's, there's so much that can affect these samples, but the Neanderthal samples, those skeletal remains, have not been put through a taxidermy process. And the taxidermy process, and especially the processes and chemicals that were used in the 1800s, were pretty damaging to ancient DNA. That's not to say that every Neanderthal specimen is outstanding, but many of the ones that have worked very well have been from colder, drier cave site and environments, and it's amazing some of the preservation that exists there. Whereas in different parts of the world, ancient DNA studies from specimens much more recent has proven really difficult to work with, including with some of our other work in the ancient DNA lab.
I think we should back up as well and say a little about what genome sequencing is, what we mean when we say we're sequencing a genome. Over the last 10 years or so, I think molecular biology in general, genetics has been revolutionized by the availability of machines for doing genome sequencing very cheaply and very quickly. So these are a set of machines or technologies called high throughput sequencers. We have several of them on campus here. And they give you the ability to read out the letters of a DNA sequence, of a string of DNA. So what you use those machines for, actually it's spread out into a lot of different types of uses that we apply these machines to do. Obviously, if we're trying to read out what the letters in some animal or human's DNA is, we can sequence their genome. So the approach for that is the machines themselves can sequence hundreds of millions of fragments of DNA all at once, very quickly, over the span of 12 to 24 hours or something like that, depending on what the machine is. And they can do them now very, very cheaply. So what took the Human Genome Project, which finished up around 2001 or so, it was going on about 20 years ago. That was a major effort, major investment in US science that sequenced every letter in the human genome, or in the average human genome, or in a representative human genome at least. So that cost about $300 million just on the sequencing side, just for the experiments that figured out the letters. And you can generate now in a lab here at Penn State the exact same number of, or the same amount of DNA sequencing data for on the order of a couple of $1,000s. But it's moved way beyond just understanding what genomes are. I mean, of course, we can look at evolutionary questions about what the relationships are between humans and other animals, or between present-day animals and ancient animals. But it's also moved into understanding what parts of the genome are serving particular purposes.
The field of bioinformatics that you're talking about and that your course covers is a pretty new field that kind of grew out of this mass of data that was being produced, right?
Well, I think the application of computational techniques to biological data has been around for quite a while. Some of the founders of bioinformatics were operating even in the 50s and 60s, in very early forms and small amounts of biological data at that time. But it's really become, the need for bioinformatics has become quite prominent given these types of machines that are generating massive amounts of data very rapidly.
Right.
So these machines are not just here at Penn State, they're everywhere. So figuring out what all that data means is bioinformatics, is computational biology, or requires computation techniques at least. No one's going to go in and figure out what hundreds of millions of DNA fragments mean or stitch them together by hand. So yes, the need for bioinformatics is growing quite a lot. I think it's a need that's there for everyone that's being trained in molecular biology right now. There's no way that you're going to go forward in a career as a molecular biologist and not need some computational techniques at one point or another. So my course here for the Intro to Bioinformatics, that's one of several courses that has been instantiated or developed over the last few years here at Penn State to serve the skills and needs of future biologists that have been trained here.
And then, PJ, can you talk a little bit about some of the specific places you got the samples for the NITTANY lion genome?
Yeah, and let me start that process by saying that what we've done so far, it's been a student-led project. Part of the idea for this project is to showcase, to provide opportunities for Penn State students to be involved in research and to showcase what they can do. And the results that we have so far in the first phase of the project, and then Shaun's class is working on the second phase of the project, but the first phase of the project was led by an outstanding Penn State undergraduate student, Maya Evanitsky, who mastered the clean lab ancient DNA laboratory work techniques, also the bioinformatics techniques. She wrote a paper, and she did a lot of outreach and talking to reporters and so on about the study, outreach to communities, to classes, and so on. So, an incredible effort that she led, and then a lot of other students were involved in. And now a lot more students get to be involved in the next phase of the project through Shaun's class, for example. So we hope to continue that. As far as the outset of the project, we crowdfunded from over 140 donors the funds to get this project off the ground in the first phase, and some of the funds for the second phase. So we had a series of outreach activities to raise awareness about the project. We had these donations come in. We had several samples that we had already lined up. And those samples were from the Penn State All Sports Museum. There's the NITTANY lion at the Penn State All Sports Museum, the Thomas Tabor Museum at the Lycoming Historical Society. The Albright College Biology Department provided a sample from a NITTANY lion that they have. The Pennsylvania State Museum also provided a sample. And in most of these cases, Maya Evanitsky, the student who led the project, she traveled to these NITTANY lions and took a small skin sample from the underside of the lion. Now, as we were doing, a neat part of this story is that as we were doing the outreach and raising awareness about the onset of the project, Bill and Carol Thomas, Carol Thomas reached out to us and said that her family had two Nittany lion skins, and that we were welcome to sample those. So it became true community involvement in terms of where the total sample size came from to kick off this project.
So what kind of things did you learn in that first phase?
Yeah, for the first phase of the study, we focused on something called mitochondrial DNA. So there are different components of everyone's genome, and one component is from DNA that's part of the mitochondria, or the powerhouse of the cell. And the mitochondrial DNA is inherited in almost universally, almost every single case, only from mothers to their children. For the mitochondrial DNA in the first phase of the study, what we found was that the Nittany lions that we studied were not different than mountain lions from the Western United States, the living mountain lions today. But again, this is only a small piece of the genome that is passed down only from mothers. So we might get a more nuanced picture when we analyze the phase 2 data from the nuclear genome.
If you're interested in like learning if the Nittany lion was a separate species, or if it had variation that was different, what are you comparing that to? I mean, you mentioned the Western mountain lions. Are those genomes available, and is that mainly what you're using as comparison?
This is one of the great things about many fields of science today, but especially genomics, is that when we publish studies, we then make the data from those published studies available for other researchers and other students to build on. So there were previous studies of DNA sequences of mitochondrial genomes from mountain lions from across the rest of the United States and in Central and South America. So we were able to download those data and compare them to the new data that we generated for the Nittany lions in this particular study. And then now we have since made the new data that we generated available to others so that future people who come along can replicate our results, but also build on them in their new studies.
What kind of things are you looking for to say this is a whole new species?
We did not think that the Nittany lion or northeastern mountain lions were a different species. It's been thought all along that they're the same species, but there's questions about how different the northeastern mountain lions were from mountain lions that still exist in other parts of the United States. So sometimes they've been named as different subspecies. Sometimes they've just been considered different populations. So what we're doing is we're taking the sequence from each of the individuals from the Nittany lions, and it's a string of letters. And if you inherit mitochondrial DNA from your mother, probably you have the same mitochondrial DNA sequences as your mother. But every so many generations, there's a new change or a mutation, basically a small change or mistake where one letter gets changed. And over time, those changes get larger and larger. There become more changes over a given amount of DNA sequence. So what we do is we compare the sequence of all the individuals to each other, and individuals with more differences probably had a common ancestor or a common relative longer ago in the past than individuals with fewer sequence differences between them. And so that's what we did. And for the mitochondrial DNA, they were very, very similar between the northeastern mountain lions and the West Coast mountain lions.
So to get a little bit technical, when you're looking at mitochondrial DNA specifically, like how confident are you in that the sequence you're seeing from the ancient samples is accurate? What kind of coverage are you looking at to get that so that you're confident enough in this DNA which has degraded over time, being able to compare that to more modern samples?
That's a great question, and especially because when I was talking about some of the challenges of ancient DNA, the ancient DNA gets damaged over time, and so the ideal thing with ancient DNA is to sequence each spot in the genome or the section of DNA that you're studying many times, so that you do become confident in the sequence, and not that you are making accidental errors in the sequence that you think that you have. So the quality of data that we obtained from the different mountain lions varied across individuals, but from some of them we had exceptionally good coverage, as you mentioned, of the mitochondrial genome sequence, with more than 100 different sequence reads covering each individual base pair position in the mitochondrial genome. So for several of those sequences, we feel very confident that we have a very accurate mitochondrial genome. And for others, there could be some errors here and there, but we are careful to only analyze the parts where we have more coverage and therefore less error.
So what makes Penn State such a good place to be doing this research?
Yeah, great question. And it's an easy answer because Penn State has a lot of resources from start to finish to be able to do an integrative, exciting, and innovative project like this. So, I mentioned before that we have an ancient DNA lab facility at Penn State. That's not something that every university has, and it's allowed us to do some really cool projects. Then also the Huck Institutes for the Life Sciences has a genomics core facility where we have the sequencing machines in this facility that have generated the data from our ancient DNA samples that then we've analyzed in phase 1, and Shaun's class is analyzing in phase 2. And then the Institute for Cyber Science has some amazing computing infrastructure that facilitates the bioinformatics work that's necessary to work with data sets like this. And again, students are getting to experience this from start to finish.
To bring this back around to the class, I mean, I think one of the coolest aspects of this project is the involvement of the students, and from the beginning of phase 1 now into the class. Can you talk a little bit more about what you've seen in the class so far and how those students get involved?
So there's something unique about this class as well, where we're not requiring that people come in with a lot of computational knowledge. So we get a lot of students coming in from B&B majors or biology majors, other bioscience majors that do not have exposure to computational analysis techniques beforehand. Similarly, we have had students come in from the computer science disciplines and majors that have not had exposure to biology before, but know that there's a big data area sitting there in biology and bioinformatics that their skills and techniques might be useful for going forward. So there's always in the class over the last few years been this nice balance between, and this nice sort of interdisciplinary relationship between the students that are coming from biology and computer science. So we tailor the projects depending on the makeup of the class and depending on which teams we can form that have a mix of those skills. However, sometimes we have biology students that are really interested in doing analysis themselves without having to learn programming or without having to gain those computational skills. One really nice aspect, one niche technology that is developed here at Penn State that makes my life a lot easier in class and beyond in research is the Galaxy project. So this is a project that has been put together by Anton Nekrotenko in biochemistry molecular biology and by his students and collaborators over many years. And it's an online system and platform for doing analysis with this type of huge genome sequencing data. So we can plug, so it really gives you a web interface to a lot of these different analysis tools that we would want to apply to these genomic data sets, such as quality control analyses, how to clean up the data and how to tell which is the high quality sequences versus the low quality ancient sequences. And then also tools in there for doing some assembly of the genomic sequences, for doing downstream evolutionary analyses, for comparing the sequences that we get out of the NITTANY lion sequences with other available sequences for other mountain lions or other feline species. So actually, just while this project was going on, again, this goes back to what PJ was saying about the culture in genomics about sharing data. The first year that we ran this, as we were running this project, I guess it was 2 falls ago, there was a group in, I believe, UCSC, Beth Shapiro's group. Beth Shapiro used to be here at Penn State as a faculty member. Her group was sequencing and assembling the genome of the modern Western mountain lion. So she was putting that data up online as we were comparing our Eastern NITTANY mountain lion DNA. So that's been a really nice resource for just, well, what should we expect, or how, are there differences in this DNA that we get from the NITTANY lion compared with the Western mountain lion genome? So as I guess we've had the ability to give students real practical experience with analyzing this type of data through these types of platforms and through these other data sets that are out there.
I know that there is one NITTANY lion, at least on campus, who is often seen running around at sporting events. Has anybody tried to get a swab of his cheek?
We have not gone down that road.
Thank you both so much for joining us, talking about this really cool project, and hopefully we'll hear more about it as things progress.
Thanks for having us.
Absolutely. Thank you.
Those guys were great.
Yeah, and it's also great knowing that we have all the tools available here at Penn State for them to sequence the NITTANY lion genome and then compare it to the data that's available on the Western mountain lion.
Right. That's super cool. I guess the only sad thing is we know that the next time we climb Mount Nittany, we won't actually be running into any NITTANY lions.
We might run into a porcupine, though.
Those are scary too, though, right?
Yeah, they're pretty fast, too.
Well, if you want to learn more about the NITTANY lion genome project or Shaun or PJ's other research, we will have links to those in the show notes below.
Yeah, so thanks for watching, and we'll see you next time.
Bye.
Clapping for audio sync. We're going to do the friends clap.
Is that ridiculous?
No, I like it. It makes me look like an idiot.
Hey, Nate.
Yeah, Sam.
Have you ever been to the top of Mount Nittany?
No, I have not. Hey, computer, have you ever got an email?
It's tomato.
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 Indeed.
Tapeworms and lice. Going on.
It's ongoing. Never ends. Science doesn't stop.
Where we talk about the research going on here. I'm going to say cool research.
You're having a
going on
Europa moment, and he uses computational. Nope.
There's some podcast about all the zestful research going on here. We'll have links to those in the
show notes. Give me show notes. That was full of zest.
Zestful, you could say. What is a zestful conversation? It's sad that mountain lions are extinct in this area. But there is one place where you can always catch a NITTANY lion in the wild, and that's at a Penn State football game.